like to thank Inbal once again for kindly accepting our last-minute invitation. And so Inbal, she did her bachelor in psychology and master in neurobiology and PhD in neurobiology and psychology under the supervision of Professor Ian Yah and Professor Benku in the Hebrew University. And then she did her postdoc abroad in Stanford, and she worked with Carl Dijkgraaf, the father of optogenetics. And now she's back to her alma mater, and she's also part of the LDAC, and she's going to show us with your green line. Hi, there are chairs here.
Okay, so I'll start by presenting the work I did at Stanford with optogenetic modulation of neurons, and then go to tell you about my plans for my future lab here with glia modulation. Okay, so as Anna said, I started at the psychology department, and I went there very enthusiastic to learn about human behavior, and then it took me about a week and a half to understand that if I want to know something about behavior and being able to modulate it in order to learn about it, I have to move to animal models. So during my PhD studies, I worked on the effect of immune proteins on the brain, especially the hippocampus, and their effect on both cognitive and emotional behavior. So I worked on depression models and on cognition and impaired cognition questions. And then for my postdoc at Carl's lab, I wanted to close this loop and start from a single cell and go all the way to the behaving animal. And in order to control specific cellular populations, I wanted to learn how to use optogenetics. So the human condition that I start from in order to follow this loop is post-traumatic stress disorder. And one of the advantages of studying this disorder is that it has a very good model in mice. So in PTSD, post-traumatic stress disorder, a single exposure to a traumatic event can lead to a lifelong behavioral problems that stem from the recurrent recall of this traumatic memory. So we have a single exposure, a single one is sufficient, though in some cases there are more, of course. And then you have a lifelong problem, and there are actually no treatments to treat the core problem, which is this involuntary recall of the traumatic experience. We can treat the side effects, but not the problem itself. And the model we can use in both humans and rodents to study PTSD is called fear conditioning. And in fear conditioning, we match a conditioned stimulus, a stimulus that had no significance in the past. We match it to a frightening stimulus, the unconditioned stimulus, like a food shock, for example. And then we can teach an animal to fear a tone, to fear a new context, to fear just about any neutral stimulus that we choose. So during the acquisition phase, we teach the animal to fear something. And then after a certain period of time, we check for recall of this memory. And it can take between minutes to years even, between the acquisition and the recall. And specifically, I'm going to talk about hippocampal-dependent fear conditioning, although there are other kinds, and I'll touch on that later. So what do we know about this transition between a memory that was acquired minutes ago and a memory that was acquired weeks or months or even years in the past? So in human studies, many studies report that there is a gradient loss of memory. And in people with hippocampal lesions, you'll see a loss of recent memories. They are unable to create new memories and to remember things from the near past. But they have no problem remembering things from their kindergarten experience, maybe, even though they can't recall what happened yesterday. And in animal models, we see a similar thing if you take a mouse and teach it to fear a context, for example. And then you take its hippocampus out the day after this memory acquisition, and then test it later, the mouse will remember nothing, okay? Because when you take the hippocampus out, you interfere with recent memory acquisition and consolidation, in this case consolidation because it's after the fear conditioning was acquired, and you will get a complete memory loss. However, if you teach the mouse to fear a certain stimulus and then take the hippocampus out a month later and then test the mouse, and you see it has no problem remembering this contextual memory that it acquired. So even in mice, we see this gradient. If you interrupt with the hippocampus at an early stage, you get amnesia. If you interrupt at a remote stage, you get no effect. So based on these findings in both humans and mice, the following model was suggested. That initially, the hippocampus is the memory manager. It connects all the different parts of the context, the olfactory part and the visual part and the auditory part, everything that makes the whole context, all the different senses. And then, as time goes by, these functions are gradually moved to the cortex, and at the end of several weeks, the hippocampus is no longer necessary. Now, when you look on this model, based on the very basic rules of neuroscience, if you look on the upper part, it makes sense, right? If these different cortical modules are repeatedly activated successfully over and over again, it makes sense that new connections between them will evolve and that they'll get strengthened. But when you look on the bottom part, to me, it doesn't make any sense, right? If the hippocampus successfully activates the same areas over and over again, by everything we're taught in Neuroscience 101, these connections should strengthen, right, not weaken. Change may be definitely evolved into something different, yes, but disappear, that made no sense to me. And when you go back to the literature, you see that the picture that I just presented about past findings is also not that simple, because in humans you sometimes see not a gradual memory loss, but a complete memory loss following hippocampal lesions. And when you look on what people do remember, the ones that do not remember what happened yesterday, but do remember what happened 40 years ago, you see that their memory is lacking. They remember the situation, but not the fine details. So you do see a gradient when you look on the finer aspects of memory. So what I wanted to do is to put this model to a test. So I'm yes.
Complete memory loss means all long-term memories are lost?
Yes, together with the ability to create new ones. Yes. Okay, so I want to ask a very simple question. How are CA1 hippocampal neurons involved in recent and remote memory and the transition between the two? But actually, this question is not that simple, right? We know the structure of the hippocampus. We know where these neurons are. We know which kind of neurons they are. We even know pretty much about the connections between these areas. But if I want to only stimulate or inhibit CA1 neurons, I have a problem because they're not the only kind of neurons around. They're not even the only, because there are inhibitory neurons and granular neurons and many other kinds. And they're not even the only cell type in this area because there are astrocytes around it, I mentioned later. So in a magical world, like the perfect world, I would like to have a manipulation that will allow me cell type specificity. I want to stimulate these cells and these cells only. And I want high spatial resolution and a high temporal resolution. And lucky for me, there's optogenetics to allow me to get to this magical option. Okay, so what's the idea behind optogenetics? And I know most of you are familiar with this, so I'll go over it pretty fast. So when you insert an electrode into the brain and you want to stimulate a specific neuronal population, again, you insert the electrode, you get the effect that you want, right, because you're stimulating the neurons that this electrode see, but you also get a side effect which you cannot control, right? Everything will be affected not even, it's not even restricted to neurons only. However, with optogenetics, we express light-sensitive proteins in a specific genetic population, right? We choose the promoter that we use to express this protein under, and then we can target specific populations. And then when we insert an optical fiber into the brain, we can get a specific effect. We're activating or inhibiting exactly the population that we want to affect, and we get no side effect because the other cell population does not express this protein, and light does nothing to it. And another advantage of optogenetics is that you can not only excite cells, but also inhibit them. So we achieve excitation with channelrhodopsin. These are cation channels extracted from algae, and they just allow cations to flow into the cell. So they depolarize the neurons and make them spike at the end. But on the other hand, we have halorhodopsin. That's a chloride pump from a bacterial source, and it will inhibit the cells from firing. So now that I have these tools, I want to get back to the question that I want to ask and show you step by step that I can actually ask this question. So we're getting back to this model. So to convince you that I can challenge this model, I have to show you that I can indeed get cell type specificity, a high spatial and temporal resolution, and actually get an effect on behavior before I even go and see if this model is right or wrong. So for cell type specificity, I made a lentivirus expressing halorhodopsin under the CAMK promoter specific to excitatory glutamatergic neurons. That's a very simple procedure. And now these neurons in CA1 will express halorhodopsin, meaning that when I shine green light on them, they will cease to fire, and I'll show you that they do. And I will have a way of shutting down CA1. So you see that halorhodopsin attached to YFP is expressed very nicely in CA1 neurons, that this expression is restricted to CA1. I'm not affecting other hippocampal regions, and that it's specific, that every neuron, practically every neuron that expresses halorhodopsin is indeed a glutamatergic excitatory neuron. Great. So the tool is working, and now the question is, of course, if it has any effect. And the answer is yes. So to test the effect, I'm inserting an optical fiber attached to an electrode into the brain, and in this case into CA1, which is the area that expresses halorhodopsin. And what I can see is I can record multi-unit activity in the CA1. You see that this area is not very active, but you do see an activity, and you do see this activity going away as soon as the light is turned on. Okay, so we have a physiological effect. And now another question, of course, great, you can see a physiological effect with the previous generations of halorhodopsin as well, but not a behavioral one. So in order to test that, I went back to the fear conditioning paradigm, and I want to explain the paradigm in more detail so you'll understand exactly what I did. So in the first stage, in the acquisition stage, we put the mouse in an unfamiliar environment. Okay, it's a cage that this mouse had never seen before, and then it hears a tone, and immediately after the tone, it receives the mild food shock. Okay, so this mouse can learn two things. We let it think about it for a while, consolidate the memory, and then we test for two things, right? Because it learned first that it should fear the tone, because the tone says a shock is coming, but also that he fears this environment, this context, because this is where he received the tones, right? He was never in this environment before, and when he got there, he got a food shock. So it has two things to fear. First, the tone, and in order to test for that, we put the mouse in a completely different environment, another new one that's very different from the environment in which it was conditioned, and sound the tone. And the mouse shows us that it's afraid by freezing when the tone is presented. And for this, it only needs the amygdala. These are the areas that marked in blue here. The other thing we can test for is the contextual memory. We just put the mouse in this context in which it received the shock, and give it no stimulus, right? No tone, no shock, nothing. It just sits in the context, and you can see that it's afraid of the context if it's freezing there. And in this case, it's not enough to have the amygdala. We do need the amygdala for that, but we also need the hippocampus. So we have a paradigm that gives us a separation between a hippocampal-dependent memory and amygdala-dependent memory. So I started with shining the light during the acquisition stage. You can see the green arrow up there, and by that I mean that I place two optical fibers above CA1, it's bilateral, and shine a light. And when you do that during the acquisition phase, when the mouse learns the paradigm, and test it a day later without light, you can see that the control group shows freezing, meaning that it's afraid of this new context. And in the halo-rhodopsin group, you see practically no freezing, almost no freezing at all, meaning that they did not acquire this task. But when we look on auditory cued conditioning, right, which happened at the very same time, we do not see a significant difference there. Now, what happens if we shine, and when I test these same mice, retrain them without light, and then test them again, you can see that they can acquire this memory very easily, and they remember just fine. So now what happens if we shine the light during the recall phase, not the acquisition? Right, these mice already acquired the task. They remember that this context is something they should be afraid of, and they show us fear. What happens if we shine the light now in the same context? So you see that these mice, after they acquire the memory, we know it's there, cease to show fear. Right, the control group, the purple group, still freezes in this new context, in this old context, and the halo-rhodopsin mice do not. Okay, they just show no freezing, so we somehow prevent recall from occurring. And this is again specific to the hippocampus, because when we test for auditory cued memory, we get nothing. They remember it perfectly fine. So we're specifically modulating the part of this task that depends on the hippocampus. So if we go back to the model, it seems that at least the first part of the model is sound. And now we can go and test the other part. So what I did to test remote memory was to train the mice and then wait a whole month, okay, for whole 4 weeks before testing them. And then shining the light during the testing, and you see that we have a remote memory task here, and we get almost zero memory in the halo-rhodopsin group. And I want to remind you that in the literature, if you do a lesion, if you freeze this area, if you pharmacologically block the hippocampus, you get perfectly normal memory when you do it a month after acquisition. And in my case, I got a perfectly abnormal memory. If I test the same mice the day after with the light off, you see that they do remember the context. It's just that when CA1 is shut down, they're unable to retrieve it. So it seems that in our hands, the last part of the model doesn't hold. But again, every other study in the literature shows the exact opposite. So in order to try and understand the difference, first we just replicated what was done in the past. So, and this result, by the way, holds in this case 9 weeks, and I also did 12 weeks. As long as you test, as long as that there is a memory trace, we can shut it off. And now let's try to do the same thing with pharmacology. So again, we trained the mice. In this case, we waited for 1 day. Okay, this is for recent memory, and injected TTX and CNQX into the hippocampus bilaterally. And as expected, we do get a memory decrease. This is for recent memory. Now, what about remote memory? Now we train the mice, wait for a whole month, inject them with TTX and CNQX, and get nothing at all, as was published before. Okay, so I have 2 experiments, the leftmost and rightmost, that are practically the same. Okay, let's look on them side by side. These 2 experiments are the same, but they give me exactly opposite results. And now the question is why? there are several differences between the optogenetic manipulation and the pharmacological ones, and I think the two major ones is first the spatial specificity. With optogenetics, I'm just targeting CA1. With the drugs go everywhere around the hippocampus. But a more important difference is these 30 minutes, right? This is just what was done in the past. You inject a drug, you let it time to travel from the end of your cannula, from the injection cannula all around the hippocampus, and then you test. And these 30 minutes, we thought, are the key to understanding the difference between these two experiments. So in order to test that, we did the following experiment. First, we repeated what we did before with another group of mice. Okay, so in this precise group, we did exactly what we did before. We trained the mice, we waited for a month, and then we shined the light on CA1 during recall and got memory impairment. This is already good. We have our internal replication. Excellent. Now we took another group of mice, and in this group, we turned the light on 30 minutes before we put these mice in the conditioning cage. Okay, so the light is on, meaning the CA1 is off for 30 minutes. And then we take the mouse, fibers still attached, light still on, everything still on, and move it to the conditioning cage and test it. And in this case, we get no memory inhibition. Okay, so these 30 minutes of light on before testing is enough to give us this strange difference. And by the way, if we take the same prolonged group the day after and then test it with the light on only during testing, right, without it 30 minutes, only during testing, then again we can get, not again, in this group for the first time, we can get memory inhibition. So now, the first way to explain it is very simple. So, well, helordopsin is not supposed to work 30 minutes without stopping, pumping and pumping and pumping. Probably the opsin got tired sometime along the way, and we don't get an effect because we don't get chloride still going into the cell and inhibiting it. So in order to make sure that this is not the case, we took slices, brain slices from mice from these groups, and these are 4-month-old mice, and injected currents into a halorhodopsin expressing cell, and this current injection in a regular condition will cause this cell to fire, but as soon as the light is turned on, the cell stops firing, and this inhibition goes on for 30 whole minutes. And then as soon as we turn the light off, the cell goes back to firing as before. So we're not killing the cells or anything like that. So if this is not the explanation, that it seems like when the brain is given enough time of hippocampal shutdown, it can somehow compensate for this and support the memory recall in other ways. And now the question is, of course, which area can support such a backup plan? So in order to answer that, we did a very tiresome experiment in which we looked on c-fos expression throughout the brain, almost in every area you can think of. And c-fos is supposed to be a marker for synaptic activity. This only holds for excitatory neurons. It just doesn't work in inhibitory ones that are fast spiking already. But when you see a cell expressing c-fos, it's supposed to tell you that this cell was recently active. Okay? So we took animals, trained them in the fear conditioning paradigm, and then tested them for their fear conditioning with either no light or with the light on only during the testing, which we call precise illumination, or with the prolonged illumination, meaning that the CA1 is shut down for half an hour before the test and during the test, and then looked on many different brain areas. And this graph is very heavy, so let's go over it gradually. So first of all, we're inhibiting CA1. So in both the precise and the prolonged case, we see a decrease in c-fos expression in the hippocampus, which makes sense. Now, if we go to the amygdala, which mediates the fear response, you can see that for the precise group, we see a very big decrease in, or there is no increase in amygdalar activity, which makes sense. If these mice do not remember the context, they have nothing to be afraid of, so you don't see any activity in the amygdala. In the prolonged group, on the other hand, these mice do remember the context, so you also see this fear response. But I can't say that this mediates the fact that they remember. This is just an expression of the memory and the fear it induces. Now, the interesting part is when we look on the anterior cingulate cortex, and let's start from the precise group. So in the precise group, we see an inhibition of this area, meaning that normally what causes this increase that we see during memory recall, what causes this, mediates this increase is the hippocampus, or CA1, and when we inhibit the hippocampus, we also inhibit the increase in ACC activity. This is for the precise group. But what happens when we shut down CA1 for a longer period of time? In this case, not only that we don't see any inhibition, but these animals not just go back to a normal level of c-fos expression, but they exceed this normal level by almost by twofold. So you can actually see this area taking over, moving from inhibition to not just back to a normal state, but you see this area actually being recruited. And what we thought is that it represents this area compensating for the fact that the hippocampus is turned off. And now, of course, thinking it is not enough, I have to show you that this area is actually involved in remote memory. And to show, sorry, yes? Yes?
Any some correlation, I know you can do it, but between the activity of the c-fos, let's say CA1, and the variant in the behavior?
So actually there. So I haven't even looked at it, and the reason is that for these histological studies, we have an N of 4 to 6 per group, and that's just not enough to look for, unless you have some crazy correlation. I haven't even looked. Okay, so if we think that CA1, that the ACC, is involved in remote memory, let's see if this is really the case. So in this case, we injected halorodopsin into the ACC and tested what happens when we shine the light both during recent memory recall and during remote recall. And what we see is that during recent recall, we get no effect because this area is probably not involved in this stage of the memory. But for remote recall, we do see a 30% decrease in memory, which suggests that there's some sort of a copy of this memory stored in the ACC, and the hippocampus can then, during remote recall, activate the ACC, extract this memory trace, and use it as a part of the whole memory. And you can see that we only get a 30% decrease here, not like a total elimination of the memory like we get when we inhibit the hippocampus, meaning that this is not the only area that supports the memory, but there are other areas as well that somehow capture a different aspect of this context. And interestingly, when we do the same prolonged experiment with these animals, meaning that we shine the light for 30 minutes before the test, we get exactly the same result. So we cannot compensate for whatever is lost here, which also tells us that probably nothing is actually stored in the hippocampus, but the hippocampus holds the ability to extract the different aspects or parts of the memory from the different cortical regions in which they are stored, like the ACC. And when you mess with this area, you cannot compensate for whatever aspect of the memory was lost in this case. Sorry? Yes, the hippocampus is is all right.
So if you would turn it off, and assuming it would have no effect, turning it off would decrease the level of reason power.
Can you repeat that? Sorry, I'm not following your...
It doesn't have any effect.
Oh, no, it's not that it doesn't have any effect.
Doesn't store any memory.
Right.
If you would turn it off, besides the ACC being turned off, you would see no power freezing.
No, I expect to see no freezing at all, which is what I get when the hippocampus is turned off, because what the hippocampus does is we have several copies, but I'm saying copies, but they are not the same, like several features of this memory stored in different areas, and what the hippocampus does is to extract all of them and build the context from them, right? And here you're just shutting down one part. When you shut off the hippocampus, you take away the ability, not the memory itself, but the ability to extract it. Okay.
How do you know that when you inhibit the hippocampus, what is lost is the ability to associate the location with the shock, the memory of the shock? Maybe the animal just doesn't know where it is.
Okay. Okay, so I do not think that what's lost is the ability to associate the context with the shock, because that's not the role of the hippocampus. The amygdala does that. And also you're right that, and this is a question that I can't answer, and I don't know if anyone can answer. The task of recall in this case is a task of comparison, right? The mouse has to know where it is now and to compare it to some past experience. So how do I know that I'm shutting off the ability to retrieve the past experience and not to perceive where it is right now? I don't. Oh, sorry.
How do you see if it's an auditory stimulus?
Oh, it's not involved in auditory perception. You see no effect on auditory cued conditioning. None.
Both of the hippocampus centers?
Yes. That's a very simple connection that the amygdala is doing between the auditory perception and the fearful stimulus. You don't need any higher areas in order to process that. It's a simpler form of memory. Okay, so if we're going back to the model, I can actually add 2 stages to this model in order to, at the end of the day, get to the same place. So we start similarly with the hippocampus bringing together all the aspects of the context. And what's different here is that we do see this new connection being formed, and of course this is highly simplified. We have other areas, as I showed you, coming into play here, but let's go for the simplified view. And we see some change in the connections between the hippocampus and the cortex, but definitely not a disappearance of these connections. And now, if we turn the hippocampus off, we see an impaired memory because there's nothing at this moment to support the memory retrieval. And then, if we give the system enough time, and here we're talking about time in the in minutes, not in weeks, then other areas can be recruited and support the memory. So we start from the start and end in the same place. The only thing I'm saying is that the original model does not show the physiological processes. It doesn't show what really happens. It's showing us the compensation because the temporal resolution of the methods that were used before were just not accurate enough. So we start from in the same place. We end up in the same place. I'm just adding 2 stages in between. Yes?
So are you proposing this model just for aversive memories or for any sort of memories?
What other kinds of... I tested it...
Aversive memory.
Not aversive memory? Not aversive memories are kind of difficult to achieve in this complexity level. We have navigation tasks that always depend on the hippocampus anyway, because spatial navigation is hippocampal-dependent, and you cannot differentiate between that and the memory recall, right? Because you need it now to navigate anyway. So any hippocampal impairment will show you an effect there.
I mean, non-aversive associations.
I haven't tested it. I see no reason why it wouldn't work, because I showed you that the auditory cue, for example, which does not require the hippocampus, is not affected. Now, most of the research was done, not just this one, but in general was done on aversive memory paradigms, for the very simple reason that they are way easier to establish. In other forms of memory, you need more than one exposure to teach the mouse to do something that's good for him. Teaching a mouse to avoid something or to fear something is very simple. Teach them to do something or like something requires many repetitions, and then you, embedded in the task, you already lose some of your temporal resolution. Mona.
What about things like conditional taste aversion, which not really require the hippocampus to take place?
Right. So I haven't done this with taste aversion, and again, supposedly it's hippocampal-independent, but supposedly remote memory recall was also hippocampal-independent. So one thing that may be achieved with optogenetics is a better definition of which tasks are hippocampal-dependent and which aren't. And there are many other tasks in which it's not clear if the hippocampus is involved or not. You have a mixed set of literature. Yes?
Why did you choose to inhibit the output of the hippocampus and not the input? Would you have different results if you block the input?
I don't know. The reason that I chose to inhibit the output is that this way, call me, this way I'm cheating, right? It's like, practically it's like inhibiting the whole hippocampus, right? If I'm messing with the output, you're right. But it also tells you that other regions of the hippocampus cannot compensate when you interfere with CA1, which is also something. And we see no attempt of them to, when you look on c-fos expression in these regions, in precise versus prolonged, there's nothing. There's no attempt of the hippocampus to somehow, I'm not blocking all of CA1, right? There's ventral CA1 as well, and I don't even touch this area, and I see no changes there.
But if we imagine that we do both experiments, and you then answer the question that was before, whether you block the perception or the memory retrieval?
I don't think I'll answer that either way, actually, because the hippocampus is not a sensory organ. It already gets something that is processed, right? So even if I interfere with the input, not the output region, still I don't think I'll be able to make this distinction. Okay, so one last thing about this. One of the hypotheses that we had was that if we can shut off CA1 and inhibit the memory, we may be able not only to prevent the memory from being recalled, but to interrupt it once it was already retrieved. Okay, so we have a memory. The mouse, for example, knows where it is already, and then it is, as soon as we turn CA1 off, it will be unable to continue to support this recall. So I'm skipping a few stages in this experiment. The important part is that we train these mice, and again, we're talking about remote memory, but it doesn't make any difference in this case. We put the mice in the testing cage. Okay, you can see that when the light is off, when the mice are first placed in the cage, they remember just as well as the control mice. But as soon as the light is turned on, these mice do not show us any recall. They stop showing fear, which in our eyes means that they stop recognizing the conditioning cage. Let's see if this is working. Yes. So we have two mice in a conditioning case, a control mouse and an NPHR expressing mice. This is in double speed, and the mice have to make these corrections with their head just for the weight of the fiber. But you see that in general they're frozen. And when the light is turned on, you see that the context changes somewhat, right? There's this, it's green, but you can't see it, but there's this green light. There is a change. You see that the control mouse still understands that it's the same context and continues to freeze, whereas the halorhodopsin mouse is all over the place. He's not showing us. Yes?
Is that mouse's eyes covered?
No.
Well, that's a very bright light. Isn't that enough to make it start moving around themselves?
Obviously not, because the control mouse sees exactly the same amount of light and it does not move. But ideally, this is actually one of the first behavioral studies that we did that actually worked with halorhodopsin. We did move to covered fibers, not because of the light itself confusing the mouse, but because you can actually, during time, condition the mouse to recognize the light as something that affects its brain and then react to the light itself. So we do try to avoid that now. In this specific case, I don't think it matters because it's the first time this mouse sees any light.
Is it possible that the light via the halorhodopsin is causing some sensation?
In the hippocampus?
Yeah, if you pass electrical current into the hippocampus, the animals will still stimulate there, they'll get excited, they'll run around.
But this is inhibition, not excitation.
Yeah, I know, but it's inhibition of a particular site neuron, which could be acting through interneurons to excite.
Yeah.
I don't think you know what that animal is feeling or thinking.
No, I don't think I can.
So rather than memory,
because there
might
might
be stirring them up.
with halo.
Yeah, sure.
Could be. Then I would expect this effect, for example, to be true for auditory cued conditioning as well, right? Because they're frozen for whatever reason. And then I turn the light on and you say, okay, you have some kind of motor response. And of course, by the way, I skipped this slide because it's not as interesting, but we've shown this light in the open field and we get no motor effect, no anxiety effect. No, we did like every control under the sun. And we don't see any non-specific effect. Okay. And now, as they say, for something completely different. And I'm moving from the past experiments to what I actually want to do here. And let's add another layer to this circle that I started with, the layer of glia cells. So we have twice as many glia cells in our brain than neurons. And what I want to do is to target these cells. They're easily targeted by genetic means, express opsins in these cells and stimulate or inhibit them, which is not trivial, and I'll explain why in a minute, and see what effect we get on neuronal activity, on normal behavior, and then on neuropsychiatric disease models. So why study exercise? The first answer is here, because they're beautiful. This is from the rainbow staining in the original paper. But there are better reasons than that. So many people are working on experiments that are related to neuropsychiatric disorders. These disorders are amazingly common in the general population, and despite the fact that people are working on it for decades, we know very little about what causes these diseases and about good ways to treat them, and this can be said for any of these separately as well. What we do know is first that there are a very high comorbidity between them, meaning that there are some mechanisms at least that are shared between these different disorders, because when you learn abnormal psychology, they tell you for each disorder, this one has 4% in the population, this is 1%, this is, and when you finish the course, you get to something like 98% if you add them all together. But the truth is that because of comorbidity, there are certain populations that will have several of these disorders combined. What we do know is that in each and every one of these disorders, and also in disorders that are not named here, we find some kind of associative abnormality. In each and every one of them. There are several studies in humans and in animal models showing some sort of disorder in astrocytes. So that may be a good target to study in order to try and better understand what causes these disorders. So what do astrocytes do? So there's a sort of agreement about the traditional roles of astrocytes, like supplying the neurons with energy, keeping a homogeneous and keeping the homeostasis in the extracellular environment, these kind of monitoring glutamate level, whatever, the traditional parts that you learn about for glia cells. But there are several studies that also show that astrocytes can directly modulate neuronal activity, but the problem is that the methodology is very limited. So these studies were done either in a slice or in culture, and you can either directly stimulate a single astrocyte, because if you just put an electrode that affects the whole region, you will directly affect the neurons, and then you can't measure anything. So you can either stimulate a single astrocyte and see what it does to the adjacent neurons, or you can use pharmacogenetics, express some sort of receptor, for example, in astrocytes, and see the effect the drug has on it. The problem is that it's never specific enough. So there are very little literature that actually shows a direct effect of astrocytes on neuronal activity. And another very big problem is that we have no axioms, right? When you think about neurons, you know that a neuron will reach a certain threshold and then it will fire. No matter, there's a dichotomy between firing and non-firing. In astrocytes, we don't have that. We're saying active astrocyte, but we don't really know what active astrocyte is. So in order to do these studies, we have to start from the very, very basic questions and then move up from there. So the challenge is to achieve specific astrocytic modulation without us affecting directly the neighboring neurons, and then hopefully get physiological and behavioral effects. And the solution again is optogenetics. If we can express opsins specifically in astrocytes or microglia or oligodendroglia, any kind of glia cells, we can then see what effect do these cells have on the neurons. So the requirements will be first to really get this specific expression, which I can also tell you that so far in all the different opsins that I tried, we got a very nice specificity. Another requirement will be to actually get a physiological and a behavioral effect, which I'll show you in a second that we can get, and at last, to show some sort of involvement in a disease model. I'm not there yet, but that's where we aspire to get. So first we have to expand the toolbox. Everything that was published so far, and everything means 3 papers, with optogenetics in astrocytes use general rhodopsin. So we take the astrocyte and we actually expose its intracellular environment to the outside, right? We allow cations to flow into the astrocytes. We know that this does not happen physiologically. It's not something normal that happens to an astrocyte. My intuition is that actually in this way we're inhibiting the astrocytes, not exciting them, but of course I still have to prove that. So I need other tools that will either be shorter or longer or more specific or more similar to the physiological ways in which the astrocytes work. For example, I can use I just love this protein, this crazy protein, engineered protein, of course, in which a guy named Rag Iron took the intracellular part of adrenergic receptors. He used the alpha-1 and beta-2 adrenergic receptors for GQ and GS coupling. So he took the intracellular parts of these receptors, but for the transmembranal and extracellular parts, he took rhodopsin. Okay, and he just copied and pasted these parts together. And at the end of the day, you get a protein in the membrane. You shine light on this protein and get GQ activation or GS activation, depending on which one you use, which is magic. So you can take this opto-GQ or opto-GS, express them as under an associatic promoter, GFAP, and get cells in which you can control the intracellular signaling without ever exposing the intracellular part of the astrocytes to the extracellular environment. So we're not letting anything flow over the membrane, we're just activating an intracellular signaling system, and I'll show you that we do get an effect with that. So the first thing that we checked was in slices to see that we can express an opsin in the astrocyte, and you can see these clouds, they look like little clouds, the astrocyte, and check how they affect a neuron that is surrounded by them when we shine light on these astrocytes. Okay. Oh, good. So these are the same astrocytes. We express an opsin in these cells and see what it does to the neurons. And we can ask several questions. First, we can shine light on the whole environment and see what it does to the neurons. And this is a recording from a neuron that sits surrounded by GQ, opto-GQ, expressing astrocytes. And you can see that very gradually this neuron depolarizes and starts firing in response to astrocytic modulation. Okay, I'm not touching the neurons. I'm just modulating the astrocyte. And still we see an effect, and we see this repeatedly. But then we can go and ask more interesting questions. For example, we know that in culture, or some people even report it in slides, if you stimulate one astrocyte, the stimulation will spread. We call it a calcium wave because it's mediated by calcium. But one astrocyte activation can affect its neighboring astrocyte. So one question to ask is, what happens if I shine light only on these astrocytes? Will only this neuron be affected, or this one as well, or even one that's farther away? I think it's a very interesting question. Of course, I don't have an answer yet. And we can measure this effect in vivo as well. As I showed you before with halorhodopsin, we can do that with associatic stimulation as well. And can we get a behavioral effect? Yes, we can. So again, these are experiments in animals that express opsins in their astrocytes, in this case around CA1. And you see that when channelrhodopsin is expressed in astrocytes, and these astrocytes are activated, manipulated, whatever you want to call it, during training, we get decreased memory. And this is one of the reasons that I think that seizure-2 is inhibiting the astrocytes rather than stimulating them. But when we use opto-GQ in the astrocytes, not only we do not get a memory inhibition, but we get a memory enhancement, if you want. And we do know that GQ is activated in astrocytes in response to neuronal activation. So maybe we can call this associatic activation.
What do you mean the memory enhancement?
Meaning that these mice remember the context better. They show us double the freezing.
Which mistake the context sometimes?
So these mice do not recognize the context very well, so they're not freezing much. These mice do remember the context, and they even remember it better than their controls, and this is why they freeze more. Okay. So the first neuronal work was done at Stanford in Carl's lab, and I got lots of help in both science and babysitting from Ro, Charu, Matt, and Janelle. And like on TV, I would like to go to the commercial part now. So I'm setting up my new lab, trying to examine the role of glia cells in neuropsychiatric disorders. Told you I'll embarrass you. So Anat and Lara already joined, and the next people to join are still in the shadow, but you're all welcome, because we have to follow everything from a single cell to the behaving animals just about every kind of skill is useful in this context, so all I need is you and new shoes. Questions?
So, in people that have hippocampal lesions, I would expect to see in fMRI higher activations of ACC. Is there any evidence?
Yes.
Learning in the cortex, either in ADHD or in other sites, should have some is the evidence for plasticity, either IEGs or immediate plasticity during acquisition?
So I haven't looked at it, and the reason that I want to try and see what's the role of astrocytes is this very long period of time. It takes half an hour for this compensation process to kick off. And there aren't many things that can explain such long duration. I mean, for a neuron, half an hour is eternity.
But that's during testing. The memory should reside in the cortex.
No, not during testing. During this time, before testing even. Oh, you were asking during this month?
The memory should somehow transfer to the cortex.
Oh, yeah, well, me and many other people wish we knew the answer for that. No, we don't know how this process occurs. It's a huge question in human studies as well. Oh, yes. But that still doesn't answer the question, I think. Yes?
Would you expect the connection between the ACC and amygdala to go stronger because of hippocampal functional lesioning? I mean, somehow, in order to be functional, the ACC, I suspect, has to be a stronger activator of the amygdala as the output
yes.
region.
I haven't looked at it, so I don't know. I can't give you an empirical answer, but also I don't know anything that I don't know of a different study that asked this question.
Do you think it's a good direction?
There could be. I mean, there's a lot of evidence to the interplay between the hippocampus and the amygdala. I'm sure there has to be. Otherwise, it cannot support the fair memory. But I haven't checked it.
So is the context perceived primarily by visual cues or?
No. In mice, I would guess mainly by olfactory cues.
So if you then have just an olfactory aversive...
To make the different, when we test for auditory cued conditioning, we put the mouse in a completely different context, and it's very different visually, but we work very hard to make it different in olfactory parameters as well, like putting it in a different room or... Yes.
But if you just have a plain olfactory aversive, say an aversive odor, do you see the exact same effect?
I don't know. I haven't tried. Maybe Adi knows, but I haven't tried doing that, like separating it. But you can never really separate a stimulus from its environment.
For instance, with the visual, would you expect to see a similar effect if you just had a visual aversive?
I would expect it to be similar to the auditory cued stimulus.
Right. So
if it's a simple visual, just a light or something like that, I would not expect it to be hippocampal-dependent. You don't need the hippocampus for perception. You just need it for combining different sensory modalities. Okay, got? I can't hear you. I'm sorry. I'm both not understanding you and not hearing you. You could say that, but it's like a hub, okay? Sorry? So, yes, so the hippocampus supports new memories, and then its role is to transfer the information embedded in these memories to other areas. But still, in order to retrieve something, the default is to do it via the hippocampus, even at remote time points. You want to call it a memory organizer? Okay. Yes. Yeah, but that's not exactly true. When you look, I mean, in mice, we only have these very gross means to check for memory, right? In humans, where you can go to the fine details, you see that, for example, a person will tell you that it remembers its kindergarten class or its teacher, but if you'll ask this person to tell a single anecdote, no matter what, from his kindergarten days, he won't be able to provide you with something like that that's rich in detail, right? It's like they remember the frame, but not the fine details in the picture, and we don't have the means to check that in mice. I mean, actually, we do. If you fair conditioning an animal and lesion the hippocampus at a remote time, and you put the animal in the same environment, you see that it remembers it, right? But if you put it in a similar environment, it's not exactly the same, it's similar. A normal mouse will know that to differentiate between these two. But the lesion mouse will lose this finer ability, not just to remember a context, but to remember in very fine details this context. Yes. So yes. No. So we put them in an open field, for example, with the light on or off. We see no effect on mobility, on the speed, or on the track length, on the percent of time spent in the center of the arena, which is a measurement for anxiety. We see no differences. Just general behavior caused by this modulation. None.
Well, it's really it's a beautiful topic, both of them. But what I want to ask, there is an hypothesis in hippocampus research that CA1 is actually in charge of what is now, and CAC is actually comparing the what is now to our previous memory. So how this projected into your results?
It doesn't. I've no way of answering that because I haven't tried modulating CA3.
Do you have any?
Yeah. I don't want to speculate. I just didn't check it. So bye-bye.